for one thing, or what, maybe what kind of, what format your reads are in. Generally, they're going to be in FASTQ format. And we have tools for examining the quality of the data in a FASTQ file. Again, a very commonly used format for your reads. And this is an example for a read that is, I think, reads where they're 72 bases long typically. And you can see that quality tends to go down at the end. This was, I think, one of my files, and something happened during the sequencing process, and that wasn't my fault, but look, the bottom fell out of the signal right at about 41 bases in. So, all that is in there. Some of the reads were fairly intact, and some of them just had a hole in it right there. Doesn't mean you can't use them necessarily, but there are ways of cleaning up the bad pieces. And that's one of the things you have to keep in mind before you start this process. You need to make sure you're working with clean data. And here's the picture that just shows an example of what a FASTQ file may look like. They're different formats, but a lot of them follow this basic design, where you have a name, which includes a number or numbers, and typically letters, and then the sequence itself for that read, and then depending on the format, more commonly these days you'll see a plus sign, which just means that it's for the same read as above, and then you'll see the quality scores in hexadecimal format, from my understanding, and then you're back to the next to a read name, and just does it over and over again. And you may have millions of reads in a file. Your files may be 2 gigabytes, 5 gigabytes, 20 gigabytes. So that's why you need a high capacity data store for storage capacity, but also for the ability to move these files back and forth, because there's a lot of information in each read file. That's one of the powers and why so many people are using this technology, because there's just so much information in there. It's all sequence information, but there's a lot of it. And the main software we'll be discussing today is very commonly used, as I said. This is the paper published in relation to it, or one of the papers published. It's the Tuxedo workflow. It's again, and you'll start out with top hat, and then use cufflinks for assembly. And just as a sample, these are the types. This is sort of a sample of what you would do at the command line if you did this on the command line. Some of you are familiar with working on the command line. Some of you aren't. Some of you may have a system where you can work already, and bow tie and top hat, or top hat. Bow tie works within top hat. But top hat, cufflinks, cuff diff, cuff merge, they're all already installed, but a lot of you don't. And part of what the iPlant cyber infrastructure gives you is a place where everything is already installed, the applications are ready to run. If you have your data in there, it's already to go. And it gives you a GUI interface, just drag and drop or click and browse, and you can start your application and do your analysis without much trouble. And I'm comfortable working on the command line. But if I'm doing an analysis, and I can, I'll often use the discovery environment for iPlant because it's convenient and it's reliable, and it provides the tools in an easy to use, well laid out fashion. At least I say it's well laid out because I've built a lot of these tools myself. So with that caveat, this again is the basic workflow, and we're going to go through this pretty quickly today. Another way of looking at it is that since we'll be looking at two different samples, in an abstract sense, condition A, condition B, we start with the reads that correspond to each one. We map them with top hat, take the mapped reads, feed them out in separate files into cufflinks, and there's a reason for doing it that way. That is to separate the conditions. I think the way some of the applications are set up, that each file is essentially assembled independently with cufflinks. And to some extent, that's because you're doing reference-based assembly. It's not de novo assembly. With de novo assembly, you need all your data together in one pot because you need as much coverage, that is information for each part of what you're going to assemble, as you can get. It's how you separate the crap from the good stuff, essentially, just by bulk information. But it's different when you're doing the reference-based assembly. The reference-based assembly, you already know to some extent what your answers are likely to be, what your transcripts are likely to look like. You may come up with new transcripts, and if they're well covered sections of genes, and they piece together different from what is seen in the annotation, then, and you have your settings loose enough so that it's not, so you're not pushing everything into the annotation information, then you'll come up with new transcripts. But here again, this is where you sort of balance your tools. You don't even need, if you're only interested in looking at known genes, you don't even need to do this part. All you have to do is map your reads to top hat, skip cufflinks, and essentially put your mapped reads right into cuffdiff. You load in your annotation information as your only bit of information that you're going to use to define what are the transcripts and genes, and that's it. You don't have to assemble anything. Yes?
So top hat will take everything and map it to the genome, and then cufflinks is actually taking anything that doesn't map to the genome and assembles the remainder?
Well, no, it assembles it, but it takes the information from top hat where it hits on the gene, on the genes within the genome. And it also compares the information that's already known about the genome and the annotation. The transcripts are there. And it compares that. It's constantly comparing. Does this look like what's annotated, or does this look like something different? If it's seeing junctions between exons that don't normally come together, if that junction information is solid, it's going to build a new transcript. It doesn't have a choice. It can't throw out those junctions. That's where the junctional information is very important. If you just had random exons, without the junctions, it may not come together so well. Otherwise, your expression information in your studies are, you know, a lot of the information is really related to just exons. You could just study what exons are being represented in my data and get some really useful information. And sometimes that may be the most honest way of looking at your data. Because there's otherwise you're pushing the information into the assumption that those exons are part of transcripts, and which transcripts. And again, there are assumptions made within the software to make that decision. There may be cases where you want to make that decision independently, and then you may want to focus on the exons alone. And you can do that, but not necessarily with this workflow. So after cuff links comes cuff merge and then cuff diff. Cuff merge just merges all your assemblies that are done independently, and at the same time, that's the main step where everything is compared finally to the annotation and brings it all together to form your final assembly. And if your assembled transcripts look like annotated ones, then it will name those transcripts according to the annotated information. And that's handy. If they don't really match up, then they'll just have an assembly name, just any in a sense, a random name. Nothing that makes sense, but it's a name to mark that this is a new transcript, and it's up to you at some point if you need to identify what it is. It may be tied to a specific gene, but it could be still a new transcript, a new isoform. And again, your ultimate result from this workflow is the differential expression information. So here, now the top hat is one of the many applications that are available, but Top Hat was designed as part of this whole workflow, and it's a pretty efficient, these days, mapper, but as I've brought up before, it's not just that it maps reads to the genome. It also will do it in a smart way. It will record, only certain mappers will map and be able to split the read across the genome. Because if you're hitting a junction, that's what you need to do. You either do what the original bow tie did, which a read will hit either one exon or another, but it can't see them both at once. Top Hat brings that information together and says, well, this read is divided across two different exons, so it represents junctional information. So it records all that information. And that's why it's good for this purpose. There are other mapping programs, and if you're not using this workflow, and if you don't have a genome, for example, to assist you, or you don't have annotation yet, then you can be using other tools like GMap or GeneSeeker to do your mapping and do it intelligently, and to in the end provide you with the information that tells you, well, there are junctions here. These are your gene structure is falling out of this mapping information because your reads are bunched up in these gene-like structures across the genome, and it will give you, in a sense, in the output, some of that information. And Top Hat does that, but more so within the context of this kind of workflow, but it can be used with other workflows too. Now, the samples that we're going to be working with today are Arabidopsis samples, and they'll be wild type, as you can sort of guess from the brief naming here, WT1, WT2. There's also high 5. High 5 is it's a type of Arabidopsis. It's not the wild type, the Columbus form, or the Columbia form. It's a mutant, a certain documented type. But these are the numbers of reads that we'll be working with in the files that we'll literally have to work with. We're not going to be able to go through the whole workflow start to finish and actually run it and wait for it, because it takes a while. So you can imagine to map 10 million reads against the genome. It can run pretty fast, but you don't want to sit here all day waiting for this stuff. And so we've already done this, as you might imagine, and so we'll give you the results. It's the main thing that you can gather from what we're doing today is you'll see how to work with the discovery environment, but also hopefully get an understanding of what you're doing with this overall workflow. And so I think we've covered enough of that. So if you look in the discovery environment, and many of you, I assume, already have your username, you'll probably want to log into the discovery environment at this point. Hopefully you can. And if you look under the various categories as they're currently set up, there's a section that's called Tuxedo RNA-seq 2, the version 2 of the newest applications, and they're better, they're faster, a little more sophisticated. And you have this list of applications there. So I don't know if you follow how to get into that, but I'm going to switch out to looking at, say I'll log into discovery environment also. So any problems logging into the discovery environment? Here we go. And as I was saying, the apps, and if you go down to under next-gen sequencing, NGS, transcriptome, profiling, Tuxedo RNA-seq 2, you'll see all the applications. But you don't have to go through all that. If you know that your first step is to use TopHat 2, well, just type in TopHat 2. And these are the applications, and TopHat paired end, that's what that PE stands for. Paired end, I guess I didn't entirely finish that thought. Paired end sequencing means that the reads, when you develop your samples for sequencing, you'll often pull out a chunk of cDNA that may be slightly amplified, but it's essentially, you often will take your fragmented cDNA, select a specific size chunk, and if you're doing single-ended sequencing, you're sequencing from one side. If you're doing paired-end sequencing, you're sequencing from both ends, but you're not just doing that randomly. The process preserves the fact that on this single read, you're reading from one end this way and from the other end this way. And that information is preserved, and the value in that is, depending on your spacing, and that's the simplest case. There are different ways of actually accomplishing this. But in the end, what that means is you have, even if your read is only this long and your piece of DNA is this long, you have information that says, well, these two were on the same piece of DNA. And since that was on the same piece of DNA, it was one piece of RNA. It was this long, and it had this sequence at this end and this sequence at that end. And so you don't know necessarily what's in between, but you know that these pieces are related, and you can tie them together in the assembly process. So that is really a lot of how people are able to extend the size of their assemblies, and it's especially important for genome assembly. Not as much for transcriptome assembly because the sequences are often smaller, but it's still helpful if you really want to get a full sense of your transcripts, but also it gives you more reliable information. If you look at only what's mapped, when you're looking at paired end reads, when both ends are hitting the genome in the same place, then you can very securely say, okay, that's where that's supposed to map. Because you have not only 100 bases here, but 100 bases here mapping at the same time to that spot on the genome. Yes?
If it's so advantageous to use the paired end, what would be the circumstances to go with the single?
Cheaper, easier to prepare. Yeah. Maybe you couldn't afford it, like me when I did my first Illumina sequencing. So, there are different reasons, and sometimes, again, you don't necessarily have to have that information, especially for RNA-seq. But, and today, even though it's sort of confusing because the files are named, for example, WT1 and WT2, that could be interpreted as two related paired end files. They're not. So we would be using this one, top hat 2 single ended. And hopefully not to take too much time on this, but because it's probably not real exciting to watch me point and click at things, but if you've never done this specifically, and also to make it clear, the information that we'll be working with, the files that we're starting with today, and the result files that we'll be cheating with, that is looking at the answers before we actually have them, are here under iPlant training, under community data. So if you look in your data window, that's where you go to look at your data that's available to you. And by the way, if you haven't already looked at this, that all Up here is Roger AD. Those are all my files, and I'll tell you, I have a lot of files in there. And community data is generally iPlant community resources, and there's a lot of stuff in here, including genome sequences buried deep in there, but they're in there. And if you share files with people, or people share files with you, the ones that are shared with you will pop up here under shared with me. You may have already seen that, but it's good to review. And if it comes up at some point, you can say if you want to share a file with somebody, like if I want to share one of my files, again, just a quick review. If I wanted to share one of my test files, I could just click this little box, click share, and this share with collaborators will pop up a little box, and I can look for collaborators, other people, iPlant users, to share within the discovery environment. Or you can create a public link. Just keep in mind that that public link is readable by everybody if you're really secretive about your data, and some people are. In any case, let's get out of my area and back to the community data. iPlant training. And all the way down here, intro RNA-seq. And input data, number 1. Those are the read files that we were talking about. Those are the files that we can put, and we learned this, I learned this last week too, that you can click on this little symbol, whatever it is, and separate your boxes. For setting up this analysis, so I'll just say RNA-seq, give it a number in case I end up doing more than one. Input data. And I think we're going to be using entirely default settings here. And since they're all single-ended files, we don't have to put them in any paired fashion. We just drag all these files in here real fast. See, it's pretty easy. Point, click, drag. These should be fairly familiar functions for you. Your reference genome. Here again, we're working with Arabidopsis. That's already listed under our reference genome list. You can use the latest ensemble information. But if we had, something that wasn't in the iPlant data store, you could put, but you could upload your own reference genome sequence, and you can put it in there. Now you also need the reference annotations. And again, you want to match it. Your annotation should always match your genome sequence. If you don't match them, then you're screwing yourself up entirely. And here again, if you had your own genome sequence that you downloaded someplace, ideally you download it at the same time the annotation if that information were available. I think we're using default settings here. I don't remember offhand if it's Fred 33 or 64. I sort of suspect it might be 64, but I don't think it matters in this case. It might be a little more accurate if we get that. And ideally, that's something you should know. But since as part of this, we didn't do what we were supposed to do, and that would put me in a bad situation with myself if I did it this way. Because, you see, I don't know I don't have the information offhand. But if I did what I was supposed to do and ran fast QC, one of the things it tells you right up front is what's the format of my read files. But in this case, for this application, I don't think it's going to make any difference. We'll just use all the defaults and hit launch analysis, and that'll go, and it'll map those reads to the genome, and that's all there is to it. Drag and drop, click and point. Yes?
Could you give us a brief kind of quick idea of the advantages of using masked versus unmasked genomes for assembly or for reference? Are there pros and cons to both, or is it
The advantage of using what?
masked genomes versus
masked.
unmasked ones?
Well, the masked genomes, I think that some applications will make use of that information, the masking information. And I think Top Hat is one of them that can. You may have to set specific options to tell it, use the masking. And the masking, if you don't know, it largely maps out and shows, well, this is all repeat information, and you may not find this very useful for accurately mapping something to the genome. On the other hand, reads can be pretty long these days. Some of your reads may be hitting some of a long repeat section on one end, but a lot of the read is in perfectly good, solid, independent information. And so knowing at least that you are in the spot where there is the repeat information on one side and the more complicated information on the other is useful information. It depends. But you may not want it sometimes if there's a lot of confusing sites like that, you know, and that certainly comes up with plant genomes where you might be looking at 85% of the genome is repeat. Maybe you want to ignore the repeats and not use that information if you don't have to.
Yeah, just one other thing is, if you do remove the repeat sequences, your algorithm can perform a lot faster. So because it doesn't spend as much time mapping some read or collection of reads to sequences that are found all over the genome.
Right.
So,
Yeah.
That's what I was figuring, but for like functional analysis where you're actually going to pull out transcripts from your transcriptome, I mean, do you find in your experience that you miss things that are important doing the mask, or is it kind of a hard sort of?
I haven't made a comparison, but I doubt if you'll miss a lot, let's put it that way.
So if you're not really that interested in, say,